Hello, everyone. Welcome to RheumNow's coverage of EULAR 2024. I've convened our panel on lupus to discuss the abstracts that we thought were of interest at EULAR 2024 last week in Vienna. We had a number of people. We had others that were going to join us but are out sick, so it's going to be just the three of us. And we're going to talk about all the cool stuff in lupus that happened at the meeting, and there was a lot of it. So our format is we'll introduce ourselves, we'll present a few abstracts each, discuss those, and then we'll do this again all this week at this same time, 5 p.m. Eastern time. We're going to have today lupus, and then after this is going to be a panel on psoriatic arthritis, and then on rheumatoid arthritis, and then finally we're going to have a rheumatology roundup with Dr. Artie Kavanaugh and I on Thursday. So I'm Jack Cush in Dallas, Texas. I'll ask my discussants to introduce themselves.
Hello, everyone. My name is Yusof. I'm from Leeds.
And Andrea.
And I'm Fava. I'm a physician scientist from Baltimore.
And part of the Hopkins Lupus Center. Folks, I think I began when we were off camera talking about what I thought were some themes from the meeting. I think you nodded when I said one of the themes was lupus and minimizing the dose of steroids, and the other one was CAR T cell. Do either of you have another overarching theme from the meeting, Andrea?
I think I agree with you. There were a few clinical abstracts that were quite interesting to see, but not as explosive as in previous meetings.
And use.
Yeah, so there was some abstract pertaining to some potential new therapies. So there were presentation about ianalumab. There were open label of anti-CD38 monoclonal antibody, daratumumab. So yeah, there's quite a few novel promising therapies as well.
Okay, yeah, there certainly are. It is a hot therapeutic area in drug development, and no better place to see those, get the first glimpse of those is at ACR and EULAR. And while that's exciting, that's also very early, and things tend to look really good early, and not especially in phase 2. And then, as I said before, phase 3 is where the rubber meets the road, and we find out how what the real long-term potential of these drugs is going to be. But let's get into it and hear what our experts think about what they liked most in the meeting. Let's start with Dr. Fava. Well, let's start with Yus. Why don't you give us your first favorite abstract?
Okay, so I'll start with the low dose use of a glucocorticoid in lupus. So essentially, there's a lot of flavor in this because since the EULAR recommendation last year, which set a target of oral prednisolone equals to less than milligram per day. So there's a beautiful study done by a cohort, Inception cohort in Italy. So the abstract number is OP0180. So this was a retrospective study of a prospective data collection. So they look into patients who achieve remission. So define remission by SLEDAI 2K equals to 0, and also prednisolone equals or less than 5 milligram per day, and also they had to be on a stable dose of immunosuppressant, including hydroxychloroquine. So what they found, they then follow these people up who met this definition of remission criteria for over 7 years. And also after when they hit that criteria, there were 74% people managed to completely discontinue steroid, whereas 26% people still remain equals to less than 5 milligram per day. So they look into the flare rate, and interestingly, the flare rates was much lower than those who completely discontinued steroid compared to those who remains on the less than 5 milligram per day. So essentially, the take-home message from the study was that after remission criteria was met, proper steroid tapering until completion appeared to be safe and was not associated with increased risk of flare.
Yeah, and again, on a lower dose of steroids, I think we talked about this offline before, that the other study was the voclosporin study on this, right? Where they patients were treated with in one study, the ALM study, they were treated with IV cytoxan or up to 3 grams of mycophenolate, and a higher doses of steroids that was weaned down. And then they compared that to the Aurora 2 study and another study they put together where they were using lower dose steroids, and the background was mycophenolate 2 grams and also voclosporin. And while the regimens were different, it turns out that the voclosporin regimen that used less steroids got to the renal endpoint faster and had less toxicity. So just another one of the abstracts kind of making this same point. And I'd like to know what Dr. Fava thinks, because I once heard his colleague Dr. Petrie say at a meeting that we may well be able to manage lupus without steroids, and I thought, oh my God, I think I've had a stroke. But really, should we be going that hard after lowering steroids, or you're always going to need some steroids?
Yeah, I was very impressed by the abstract. I mean, also because we do not have an explanation as to why people flare more on prednisone. I think that one of the striking findings is, I mean, as expected, is the damage accrual they showed at 2 years, where they showed that basically there was a 17% damage accrual in the people who remained on prednisone versus 7% of people who tapered. But the damage accrual that the group of prednisone had was for a large part from prednisone, but there was also a part that was independent of prednisone, which was quite interesting. So I think that there is a nice way to come off prednisone, and then we should always strive for it. And I think that eventually we will get it done. There's a beautiful study that, I mean, that was highlighted in one of the sessions about in the What's on the Horizon, and one of the initial plenaries coming from Mayo, from Ali Duarte, and where they basically did a meta-regression of multiple trials of all the different kinds of steroids in lupus nephritis, plus or minus the pulses. And one of the major findings is that, well, if you pulse ahead, you have a higher chance of response in any way. So I think this resonates with some of the practices that we tend to have, at least here, where if there is a bad flare, we'd rather like hit hard with the pulse and then put patients on a very low dose of prednisone right after, instead of starting these very long regimens with 60-80 milligrams and going down over months. So I think that a steroid, at least a low, if not free, future is coming.
Yeah, I've done a lot of lupus over the years where I treat, but I'm known more for RA. But in RA, steroids are what? They're the drug that gives you rapid control of your situation while you're waiting for disease modification, right? And steroids don't really play a role in disease modification, really at all. I mean, you'd have to work hard to find these studies where you can make that point. But there's a greater reliance on steroids in lupus, because the inflammation is different, the organ damage is different and much scarier, and hence the reliance. And so this shift, as we have more therapeutic targets that we can effectively engage with, means that we should be using less steroids. So I like this progression. I think it is going to make rheumatologists better at what they do, and patients have safer outcomes in the long run. You can final say on this.
Yeah, sorry, can I just add something? So I've just got some important points as well. It was steroid was initially used in the field of lupus exactly 75 years ago. So it's nice to see, we're taking a long time to reach here, and hopefully we're nearly there.
Right.
And I think when we talk about steroids, we should make the important distinction between oral steroids from parenteral steroids, because most of the studies showing the damage coming from steroids come from oral prednisone, so oral steroids. Whereas usually we see much less of an effect with IV or even intramuscular triamcinolone, for example, that we tend to use more frequently to treat mild to moderate flares.
Yeah, I agree. All right, let's Dr. Fava, you give us your first favorite of the meeting in lupus.
Yeah, I mean, I really liked the abstract OP0089. That was a phase 2, the data on the phase 2 trial on ianalumab. This is a monoclonal that targets BAFF receptor and is engineered to be basically B-cell depleting, which is quite interesting to see because we think of B-cell depletion when we think about targeting CD20, CD19, and this is like targeting B-cell from a different receptor, which is basically the same family that's been targeted by belimumab, but this is done in a way that really depletes B cells as opposed to belimumab. And so basically, this is an extension. They did a first 28 weeks was a placebo controlled, and then there was an open extension where the patients who got placebo eventually got the drug, and it was the curves were very nice. The primary endpoint was a combination of SRI-4 and the ability to tapering steroids below a certain threshold, and there was a very nice response at week 28. But what was nice to see is that the patient that eventually got ianalumab in the open extension by week 52 caught up with the patient who'd been getting the drug the whole time. So it seems that it's going toward a medication that is targeting a meaningful pathway, is showing an effect. And so it will be very nice to see another, basically another medication that will help us targeting these B cells that now we're getting from multiple lines of evidence, the idea that these are truly driving part of the disease, because when we target them, the disease gets better and sometimes can even get cured if we get it, if you see the data on CAR-T.
So what is the extent of depletion that we see and for how long with this?
I forgot this. It was not on the abstract. They showed in the slides. I would have to think about that. I think they achieved basically the same depletion you would get with rituximab by flow cytometry. They showed that the cells went down to 0. I forgot the longitudinal data, but they really showed depletion, which was quite impressive.
Yeah, I think I agree. I think they showed that data in the ACR 2023 last year, that they had a profound depletion in the ianalumab group. Yeah, and it's quite profound because of they have a dual mode of action, so they kill the B cell through the antibody dependent cellular cytotoxicity, as well as the BAFF receptor inhibition, so inhibition B cell survival. So this dual mode of action is quite potent in terms of B cell therapy mode. Yeah.
So theoretically, that's the advantage over rituximab, which that it has a different MOA and it may have different kinetics as well, and that gives it maybe potentially more hope than what we saw with rituximab. Again, I always like to remind everybody, you may love rituximab in the condition that it's not approved for where it failed, and yet you are loath to use it where it actually has been approved in rheumatoid arthritis because you have many other choices in rheumatoid arthritis. But yeah, I mean, rituximab does work in hematologic indications, I mean, ITP and autoimmune in my experience, and maybe some select, but there's still a heavy reliance by rheumatologists on rituximab. So these newer, molecules, belimumab certainly, and now ianalumab and obinutuzumab as well. These are going to be, if you were happy with rituximab and what it may do for your lupus patients, like you're going to really be happy with these in the future. But again, we've got to get over some major phase 3 data before it's going to be FDA approved. But it's certainly, everyone that I know that's looking at this data is really encouraged. So. Okay.
And also with the positive data on Sjogren's, right? So this is one of the few biologic that targeting the immune system in Sjogren's showed like a positive trial also on Zika symptoms, which is quite impressive.
Right. Very, very hard to do. Sjogren's is where all biologics have failed thus far until this most recently. So that's also encouraging. I'll bring up the sub-analysis of the Paisley study using the TYK2 inhibitor, deucravacitinib. That study was previously presented in November 2022, and they're rolling out sub-analyses of that, and they have a larger phase 3 that's in progress in this study. In this study, they OP0448. They looked at 101 patients with chronic cutaneous lupus and looked at the deucravacitinib responses as measured by the CLASI 50 and the CLASI response criteria, and using what looks to be the right dose, which is 3 milligrams or 6 milligrams BID, about a 60%, 65% CLASI 50 response, and a CLASI 70 response at the 3 milligram BID versus placebo was 65% versus 8%. Because they were mainly treating a variety of different skin disorders. It was not only acute LE skin disease, but also discoid and subacute, and that I think ill-defined variant of chronic cutaneous LE. But and of course there was nothing else about this this study. What I find interesting about this is, number one, I think this is the way lupus trials should be going, which is having a specific end organ primary endpoint as opposed to an SRI-4, SRI-6, which is really hard to get an FDA approval on. But the problem, I think, is with the regulatory agencies and their acceptance of a skin endpoint. I don't think that that's been worked out with the FDA. So using what most researchers are saying is the best and most validated tool, the CLASI response criteria, that the FDA hasn't quite given its stamp of approval to that, and that's why you're not going to see, lupus approved initially for a cutaneous indication, and I think that's a shame. But either of you have any impressions about this sub-analysis?
Yeah, thanks for bringing this abstract, Jack. I find it really interesting as well, because as we know, cutaneous lupus is variable and also can be very difficult to treat. We've used a lot of rituximab in the UK, and we found that in terms of skin response is also variable, particularly people with chronic discoid lupus. None of them responded to the rituximab that we gave, so we are stuck. And they usually have anti-Rho antibody and no low complement, so we can't use belimumab. So we did publish before a case series using anifrolumab, and it did work really tremendously in a variety range of cutaneous lupus. So it's good, if we see the same signals again for deucravacitinib, so we got this option as well for those patients, because these are all disabling features of lupus.
Yeah, well, the comment, I think that this is another piece of evidence that when you target type 1 interferon in lupus, skin responds beautifully. The rest of the systems, it's not always the case, but in the all trials that did not complete like the other monoclonal blockers of type 1 interferon, and now we see with anifrolumab, and then now with the small molecules like deucravacitinib that target like the type 1 interferon signaling, we do see the skin response beautifully. So I think that's going to be the thing that we're going to be seeing. And then to get back to your comments about the outcome measures, this is absolutely true, and is one of the major issues in lupus trials. And I wanted to give a shout out to the major effort that Eric Morandis is taking on redefining the clinical outcomes in lupus, starting basically with what's needed from regulatory agencies in order to approve drugs, and then working with patients also to combine what's important to them, and combining basically new ways to establish response that are tailored to accelerate the approval processes. And it truly is a massive and beautiful work, but I think it will have a tremendous impact.
Yeah, it's I can tell you, I was on the FDA advisory panel for, I think, the very first advisory panel they had, trying to look at endpoint measures in lupus, and they had all the big lupus mavens around the table, and it was a difficult time back then. This was like maybe 15 plus years ago that they did this. And it still is a challenge. You know, I mean, once the FDA has its teeth into, you know, endpoints that a drug can be judged sufficiently, then drug development proceeds. And the FDA had to go through a number of these meetings, both with the advisory panel and then experts and then the companies themselves, before they got around to actually understanding how to best do the trials that were going to get approval. But I think, you know, renal endpoint's pretty easy. You know, it's, you know, I think that's already been decided upon. It's going to be harder when you start looking at, you know, non-renal or not in the general lupus. So, you know, hematologic lupus or neuropsychiatric lupus, you know, pulmonary lupus, articular lupus should be easy, but an ACR20 would probably be applicable here. But anyway, this is what we need to further develop better therapies to manage our patients in lupus. All right, our next round. Go ahead, Andrea.
If I can add one quick thing, we could go to an abstract actually that I forgot the name of the abstract, but it was basically a post-hoc analysis of the phase 2 nifrolumab study in lupus nephritis, which was a failed trial by proteinuria. And this was about checking the CD163 in the urine level, so urinary biomarker. And this is a biomarker that correlates with histological activity. And what was found is that if you look at the responders, both to placebo and anifrolumab, well, they all had a decline in CD163, which usually reflecting resolution of inflammation in the kidney. But it was interesting that when you looked at the non-responders by proteinuria, you would see that the patients who got the placebo had the flat line of CD163, and the patients who got anifrolumab, despite being non-responders by proteinuria, had a decline of CD163, at least early, which is telling us that perhaps, yes, you give a drug that is attacking the immune system, is an anti-inflammatory, you know, and generally, and then you do see an immunological response that perhaps is meaningful. We still don't know if it's meaningful or not if you don't resolve proteinuria, but I think it's a start to tell us that if we have a better treatment endpoint and outcome, perhaps we could capture this better responses.
Yeah, and this is where, you know, your biomarker work has been really, really important. And I think it's going to be not only clinically applicable, but it will be an important endpoint in clinical trials going forward. So, okay, use.
Yep. So I think we've got to talk about CAR T cells because they're everywhere at EULAR 2024. So I think if you don't talk about it, you know, you might have not attended the EULAR. But they're all early phase trial and all open label in a phase 1. So it's not randomized yet. So the one that striking a little bit, which was presented at the abstract session, was about compound CAR T cells. So the abstract title OP0017. As we all know, CAR T cells took center stage 2 years ago when Professor George Schatz presented the data on 5 patients with refractory lupus. So now 2 years, you know, forward, rather than just CD19 CAR T cells, they presented data on compound CAR T cells. So essentially, what happened during the apheresis? So the T cells were then re-engineered, not just for CD19, but they also add one more receptor, which is the BCMA. So BCMA attached on the surface of cells, including the plasma cells. So basically, it will be effective to remove the auto antibodies. So what happened? So this was open label phase 1 studies in China of 12 patients with refractory lupus nephritis. This patient had a biopsy positive prior to starting the CAR T cells, and they failed treatment, including cyclophosphamide as well. Of all the 12 patients that were treated with this compound CAR T cells, most of them achieve a complete renal response. I think there's one or two that didn't hit the spot of complete renal response. And in terms of general safety, because people are concerned that if you, with the risk of infection, particularly, I think there was one urinary tract infection recorded, and mild COVID cases as well. So the only thing was the follow-up was quite short. It was only 12 months follow-up. We do need longer follow-up for these kind of studies to see once the B cell have repopulated, whether do you have a disease relapse and so forth, and what else would you do if they do relapse? I mean, would you want to give another compound CAR T cell, which potentially going to be have a high degree of toxicity and also be expensive, or there are also other alternatives, because one of the main principle of CAR T cell is profound lymphodepletion. So we do have other alternative. For example, a better rituximab, like you were saying, Jack. If the obinutuzumab might be licensed in the future, ianalumab going for phase 3, and potentially daratumumab, which is like anti-CD38 monoclonal antibodies. So there are quite a few other alternatives there, and also potentially something that we're gonna look out in the next maybe ACR or next year. Just to be aware of another strategy called BITE, which is bispecific T cell engagers, for which there was data that shown for refractory rheumatoid arthritis in this conference. But I heard they've already done it in lupus, so we might get a result of that in the next two conference probably. Yeah.
Andrea, what's your view of this new wrinkle to CAR T cell therapy?
It's very exciting. I mean, I really look forward to seeing the results on BITE, because they really capture the same mechanisms of CAR T, and and also I think that with CAR T the gamble is to see how low we can get in terms of complexity and cost, and being off the shelf and having a low risk, to the point that we can make it available for more patients. It truly this becomes a curative treatment. And so the more effort that we get to get these new compounds that are being prepared, that are getting closer and closer, closer, there are already trials on off-the-shelf products. I think this is going to be incredibly exciting.
The one, so I heard Garrett Shett talk about 2 or 3 weeks ago at Yale. He gave a great overview of his 15 patients, 9 lupus patients, and that experience, and talked about the B cell depletion and how it comes back, and how it comes back is more, I don't know, a kinder, gentler B cell, and not quite so angry. And it makes me rethink, the pathogenesis of lupus, especially since CD19 is not found on plasma cells, and CD19 is not found on plasmacytoid dendritic cells, a major source of type 1 interferon. And up until this CAR T cell story, I would have said most of the damage in lupus is plasmacytoid dendritic cells, and what's going on at the plasma cell level, not the pre-B cell in the marrow, and not circulating B cells. And this whole, his therapy blows that out of the water. Now, this compound that Dr. Yusof's talking about, that's BCMA targeting as well. BCMA is targeting plasma cells and plasmacytoid dendritic cells. So it'll be interesting to see what the contrast is going to be, not just in the short term where it looks like there's some benefit, but also in the long term as far as benefits, but also as far as safety. So this is an exciting area. There's a whole bunch of brand new companies getting into this, all making their version of the same, using autologous and allogeneic CAR Ts as well. So it's going to get real complex real quick in the next 2 years, and I think it's going to be an exciting area. All right, Dr. Fava, you have one more that you want to review?
I do. Let me put it up here. It is so this is not about treatment. It's a diagnostic. It's after the OP0255, and was presented by Melissa Moreau from OMRF. And this is, I mean, we often see this newer like diagnostics in lupus. This was a serum-based assay where basically they measured in the I think it was serum. Anyway, it was in the peripheral blood. Multiple biomarkers, and they wanted to find a panel of biomarkers that could predict active lupus as defined by SLE. And then they drilled it down to 10 of them, and they validated it. And basically, the bottom line is that they had a panel that, with a decent amount of reproducibility, could predict in a validation cohort patients who are have active lupus as to patients who are quiescent, but also with several of these biomarkers that could identify renal lupus from non-renal lupus. Now, I'm usually very skeptical of these biomarkers that predict something that I could get for myself at the same time, right? I mean, why would I need a biomarker to tell me the current SLE if it's something that I can calculate myself? I mean, I want a biomarker that could predict if the patient is going to respond, or can predict if something is going to happen in the future. But I think that if we look at this into the broader context, for example, not everyone, especially in an area where there's not a rheumatologist available, or with with a shortage of rheumatology, arming other providers with the ability of differentiating lupus activity from something else, and perhaps catching these patients, or just more accurately identifying lupus activity as defined by a lupus expert. This lead seems easy, but it's not that trivial. I think it can provide some use in the future as this biomarkers are getting validated.
So biomarkers are a very hot area, and I like it. I think rheumatologists have become very comfortable with the biomarkers they use. And you guys, being lupus experts, tell me about the biomarkers that we really hang our hats on, and why we shouldn't be so comfortable with proteinuria and complement levels and lymphopenia as my targets for therapy.
You want to start? Use.
What do you think?
Yeah, so I think sometimes there is some discordance between the clinical pictures. For example, you do see some people with high anti-double-stranded DNA titer more than 300, but clinically they're quiescent. So so sometimes the biomarkers all these current immunology tests are good for diagnosis and not necessarily for assessment of disease activity or flare prevention. Hence, we do need better biomarkers in order to better personalize and stratify our patients. So there's quite a few other blood biomarkers. For example, we've been working on interferon biomarkers in leads, and also when we try to talk about interferon biomarkers, it's still not being used in a clinical practice, because when someone say, oh, I have a high interferon gene signature, what do they mean? What do they measure? How many interferon gene signature that you captured, and are you measuring protein or gene expression? So there's quite a lot of this variability that need to be harmonized. And certainly Andrea probably agree. I think Andrea been measuring quite a lot of urinary biomarkers, so I think there's quite a lot of a lot of validation that needed. And also, once the process validation is happening, I think we really need to focus into bringing these biomarkers into clinical practice. And as we all know, there's quite a lot of multiple hurdles to get that into practice.
Yeah, I agree. And to get to your point, Jack, about the biomarkers that we have, especially thinking about extrarenal lupus, where perhaps it's a little bit more challenging, there's much heterogeneity in lupus patients, to the point that, sure, if we have a patient with symptoms, have a rising double-stranded DNA titers and a dropping C3 and C4, well, that's, in a way, it's easier, right? but if we have the patient who never had double-stranded DNA antibodies, or the patient who never like dropped complement, we have these patients, and identifying flares in this patient may be quite challenging. And so if you are in a lupus center where we get to see the variety and the volume of patients to make us more confident, that's one setting. But in other setting, when that becomes more challenging, sometimes doctors are left with no biomarkers to go by, and yet the patient have lupus. And so having something comprehensive that can help us understanding the underlying inflammation that is more specific to lupus, it could help. It could help when what we have it's not perfect.
Yeah, I think it's I'm looking forward to this with a great deal of hope, because all the data on the newer biomarkers, the CD163, 19, the ones that you reviewed here, I think this is an exciting time for lupus management. So, and I think I always said, as I said before, it's going to change clinical trials as well. I want to ask the both of you to comment on one. There were I think there was 2 abstracts on obinutuzumab, the anti-CD20 monoclonal antibody, and the reanalysis of the phase 2 Nobility trial. The one that I saw looked at subset analyses, and again, this is the preferred B-cell depleting antibody in lymphoma, and mainly because of its characteristics and safety profile and kinetics and that it is superior to rituximab. And that's the feeling going forward in the lupus trials that have been thus far done. In this study, they showed that, again, the B cells are depleted for a year, but that when they reconstitute, it's the naive cells that come back, and the memories, I think, stay depleted longer. But what's your view of the data so far? What was presented at the meeting on obinutuzumab?
Yeah, so I did interview Dr. Vital, which is my colleague during the conference. So he presented this work. So it is post 0529. So that's the abstract number. So essentially, this was looking in terms of longitudinal biomarker assessment, and as what you said, Jack, earlier, so they found that people who achieve complete B cell depletion had a better renal response than those who had incomplete depletion. So when they mentioned this, so the degree of the B cell depletion was measured using highly sensitive flow cytometry technique. So this is different than the conventional cytometry technique. So the difference between the two essentially is the enumeration of the plasmablast cells. Because using the conventional cytometry technique, so usually the gating started on the CD19. However, as we know, the plasma cell not all exclusively come from CD19. So you have to start your gating from monocyte and multiple gating strategies. So basically, this showing in terms of the property of the type 2 anti-CD20 monoclonal antibodies through ADCC a mechanism of action that they're able to result in complete depletion. But also not just that's the first phase of it, but secondly also they were looking in terms of those who had this sustained depletion. They also had a better response as well. And then clinically, I did a lot of this work during my PhD. We did see some patient who developed anti-rituximab antibody in the subsequent cycle of rituximab. So what happened back then, we did manage to get some compassionate use of ocrelizumab. So ocrelizumab was promising initially in the trial, but then it had to be terminated because there's quite a lot of fatal infection in Asia. So what happened when we use these in the 5-hour patients? So most of patients really had complete B-cell depletion, and these patients also will come off medication. They'll come off mycophenolate and stay on hydroxychloroquine only. And when I check serial B-cell, the plasma cell still has not come back after 4 years. and then they relapse after 4.5 years. So, it was got really astounding amount of depletion that we can achieve back then. But, it's showing to say that, the importance of achieving this deep B-cell depletion to get a good and prolonged response. Yeah.
Yeah, I mean, it's hard to top that since that was your PhD use. I think it's quite concerning to, we usually, I mean, we often like rituximab, but we always prescribe it with the concern that, okay, I'm giving something that is going to immunosuppress this patient for 6 months and I can't hold it if something goes wrong. And then if we think about obinutuzumab, and now you can deplete B cell for as long as 4 years, well, that's quite concerning. And we do know that from clinical experience, these patients are, the rates of developing hypogammaglobulinemia, it's there. So I think that is a powerful new drug, but as we get to understand it better, we should really think about the long-term implications of removing an important part of the immune system.
Okay. I want to ask you your opinions also about this abstract. I think it comes from Toronto about responses in patients with membranous lupus nephritis versus proliferative lupus nephritis. It was a retrospective study from their lupus cohort, 200 patients and 50 with membranous disease, 164 with proliferative disease. And the bottom line was that it looked like patients with membranous disease deserve aggressive treatment. Were you guys familiar with this abstract?
I'm pulling it up.
It was POS 0732. Again, I thought it was interesting in that its conclusion that membranous is not just proteinuria only, that it's not a benign disease and may be associated with significant deterioration and even death rates. And on that basis, so it really was an outcomes, I think, comparison from a single center. And I have a pretty aggressive lupus clinic at the Western Hospital in Toronto. And if you look at they show Kaplan-Meier plots on all kinds of outcomes, and boy, they're very overlapping between membranous disease and GN and proliferative GN. And that kind of goes against my training that, membranous doesn't always need to be, don't give them Cytoxin. Nobody does that, whatever. And that you should be, that you're only going to treat them if their renal disease progresses or if their proteinuria is out of control. And so anyway, I thought this would challenge the conventional wisdom in lupus management.
I agree. It's very interesting. We published from the Hopkins lupus cohort a paper a few years ago in Journal of Rheumatology where we looked at the outcomes of patients with who had pure membranous on the second biopsy. So meaning you have a patient treated and then you have a rebiopsy for cause and then you find pure membranous and then you say, well, sure, it's just membranous so I can get reassured. And then what we've seen is that actually there is a significant rate of CKD progression to ESRD and including death. And so what we found is that one of the predictors of poor outcomes was having had a history of proliferative lupus nephritis in one of the previous biopsies, which is with the notion that these patients, so it's very well known that lupus nephritis patients, if you biopsy multiple times, they can go all over the place. They start at 5, they go to 4, then they go to 2, and they back to 4. So they can do whatever they please. And so, yes, this patient may be membranous at the time of biopsy, but there may be a kinetic problem as it was described in one of the talks, that was this kinetic chaos, that maybe this patient is turning to proliferative after a while. And we don't know because we can't just rebiopsy patients every few months. And this is also an area where urinary biomarker can really help. So I think that it is good to see that this patients with non-class 3 or non-class 4 lupus nephritis are getting more attention because things may change. The other thing I would like to add is that when we studied pure membranous lupus nephritis in terms of their proteomic profiles, we found, surprisingly, that a lot of these patients had signature of inflammation, such as neutrophil degranulation and also extracellular matrix degradation. What? We say, oh, this is reassuring, it's class 5, and we see active degradation of the matrix. It's not something that we have in mind with that phenotype. So, and there's an abundance of new data also coming from class 2 lupus nephritis that is showing that that perhaps is the gateway and is to then class 3 and class 4, showing that perhaps it's more aggressive than we thought. And so I think that seeing lupus nephritis is kind of like a more heterogeneous group of diseases that is also like kinetically diverse should be in our mind when we treat these patients.
Do you have a different view on this?
Yeah, so I think there's also been some abstract presented in ACR. I think last year. I think we're looking into the just post hoc analysis of the voclosporin trial. So although it was not powered to answer the question, but one of the question was asking whether it is effective in people with class 5 membranous lupus nephritis. And what they found, the response using voclosporin was as good as treating the class 3 and 4 as well. But this is likely because of the preservation of the proteinuria, because that's what where the voclosporin works. And I do find in my clinical practice as well, and when I do see a biopsy of like class 5 active membranous membranous lupus nephritis. What we found, I tend to use the voclosporin first, or cyclosporin first, because just to conserve the heavy leakage of proteinuria before I start using different immunosuppressants, because prior to the advance in cyclosporin development, when we use a lot of like rituximab, we found that potentially nothing happened. It might have been like you give rituximab and it just speed it off after the infusion. So I think that's what how we practice these days as well.
Okay. All right. I think that's plenty to digest in lupus world. I want to thank Dr. Fava and Dr. Yusof for contributing their time and spending and focusing on lupus at EULAR 2024. And we'll be doing it again at ACR in a few months. So thanks, everyone. Have a good night.
Okay, bye-bye.
Thank you. Good night.